Hey everyone! Welcome to the Ncert Audiobooks Library, the best place to listen and learn from Ncert textbooks. Whether you are preparing for exams, revising a topic, you can find the audio version of any Ncert textbook here.
Sexual reproduction in flowering plants. Flower, a fascinating organ of angiosperms. Pre-fertilisation, structures and events. Double fertilisation. Post-fertilisation, structures and events. Apomixis and Polyembryony. Chapter 2. Sexual reproduction in flowering plants. 2.1 Flower, a fascinating organ of angiosperms. 2.2 Pre-fertilisation, structures and events. 2.3 Double fertilisation. 2.4 Post-fertilisation, structures and events. 2.5 Apomixis and Polyembryony. 2.1 Flower, a fascinating organ of angiosperms. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swoon over, the rich colors that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structures of the inflorescences, flowers and floral parts, shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure and the processes of sexual reproduction in flowering plants, angiosperms. 2.1 Flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious and cultural value. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of 5 more flowers that are used in social and cultural celebrations in your family. Have you heard of floriculture? What does it refer to? Figure 2.1 A diagrammatic representation of LS of a flower. To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. In Class 11, you have read the various parts of a flower. Figure 2.1 will help you recall the parts of a typical flower. Can you name the 2 parts in a flower in which the 2 most important units of sexual reproduction develop? 2.2 Pre-fertilisation, structures and events. Much before the actual flower is seen on the plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a whorl of stamens representing the male reproductive organ, and the gynoecium represents the female reproductive organ. 2.2.1 Stamen, microsporangium and pollen grain. Figure 2.2 shows the 2 parts of a typical stamen, the long and slender stalk called the filament, and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus, or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed with each lobe having 2 theca, i.e., they are edithecous. Figure 2.2b. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther, figure 2.3a. The bilobe nature of an anther is very distinct in the transverse section of the anther. The anther is a 4-sided tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers, figure 2.3b, the epidermis, endothecium, middle layers and the tapetum. The outer 3 wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than 1 nucleus. Can you think of how tapetal cells could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrad? A, B, C. Figure 2.3a, transverse section of a young anther. B, enlarged view of one microsporangium showing wall layers. C, a mature dehisced anther. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad. Each one is a potential polliner microspore mother cell. The process of formation of microspores from a pollen mother cell, PMC, through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells, the microspore tetrad, figure 2.3a. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains, figure 2.3b. Inside each microsporangium several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther, figure 2.3c. Pollen grain. The pollen grains represent the male gametophytes. If you touch the open anthers of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, sizes, shapes, colors, designs, seen on the pollen grains from different species, figure 2.4. Figure 2.4 scanning electron micrographs of a few pollen grains. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent 2-layered wall. The hard outer layer called theoxina is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exene has prominent apertures called germ pores where sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The exene exhibits a fascinating array of patterns and designs. Why do you think the exene should be hard? What is the function of germ pore? The inner wall of the pollen grain is called thentine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains 2 cells, the vegetative cell and generative cell, figure 2.5b. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this 2-celled stage. In the remaining species, the generative cell divides mitotically to give rise to the 2 male gametes before pollen grains are shed, 3-celled stage. Figure 2.5a, enlarged view of a pollen grain tetrad. B. Stages of a microspore maturing into a pollen grain. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses, figure 2.6. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. You may have heard of storing semen/sperms of many animals including humans for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen-1960C. Such stored pollen can be used as pollen banks, similar to seed banks, in crop breeding programs. 2.2.2 The pistil, megasporangium, ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one, the pistils may be fused together, syncarpous, figure 2.7b, or may be free, apocarpous, figure 2.7c. Each pistil has 3 parts, figure 2.7a, the stigma, style and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, locule. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you studied in class 11. Arising from the placenta are themigasporangia, commonly called ovules. The number of ovules in an ovary may be 1, wheat, paddy, mango, to many, papaya, watermelon, orchids. The megasporangium, ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule, figure 2.7d. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. Opposite the micropular end is thecolaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryosausaur familigametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell, MMC, in the micropular region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of 4 megaspores, figure 2.8a. Figure 2.8a, parts of the ovule showing a large megaspore mother cell, a dyad and a tetrad of megaspores, B, 2, 4, and 8 nucleate stages of embryo sac and a mature embryo sac, C, a diagrammatic representation of the mature embryo sac. Female gametophyte. In a majority of flowering plants, one of the megaspores is functional, while the other 3 degenerate. Only the functional megaspore develops into the female gametophyte, embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. What will be the ploidy of the cells of the nucellus, MMC, the functional megaspore and female gametophyte? Let us study formation of the embryo sac in a little more detail. Figure 2.8b. The nucleus of the functional megaspore divides mitotically to form 2 nuclei which move to the opposite poles, forming the 2 nucleate embryo sac. 2 more sequential mitotic nuclear divisions result in the formation of the 4 nucleate and later the 8 nucleotistages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed immediately by cell wall formation. After the 8 nucleate stage, cell walls are laid down leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of cells inside the embryo sac, figure 2.8 BC. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropular end and constitute the egg apparatus. The egg apparatus, in turn, consists of 2 synergids and 1 egg cell. The synergids have special cellular thickenings at the micropular tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. 3 cells are at the chalazial end and are called the antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac, at maturity, though 8 nucleati is 7-celled.
The remaining portion will be covered in next audiobook. Don't forget to like, subscribe, and comment down below.